Hello, this is Mr. Buss, and in this video I'm going to walk you through the in-class activity we did in AP Biology called the RNA and proteins cutout activity. And so what we did is we started with, at each table, a DNA template strand. So these are DNA nucleotides like guanine, thymine, cytosine, adenine, and so on. And this is one strand, so of course you know DNA is double-stranded. So the complementary strand on this side is not being shown. So I'm just using one strand of DNA, knowing that you understand that there's a second strand, because when we make RNA, or when our cells take DNA and turn it into an RNA sequence, it only uses one side of that. So one side of DNA codes for RNA. So this is the DNA template strand, and it was kind of a longer strand. It actually had like one piece of paper cut out into four pieces, and so it's like, you know, four-foot strand basically. So there's a lot of A's, T's, G's, and C's. And what we did is we started by saying, where's the promoter region? And so on one of the ends, there was a TA promoter region that we looked for. So it doesn't look like it's on this end, even though there's thymines and adenines. It's not in the specific TA. And so if I look on this side, starting here with the C, then it does go TA. Now, just so you know, like that promoter region, it is more complex than that. It's actually TA, WAW, where W is like A or T and stuff. So don't worry about that. It's not a big deal. But just know that there is a TA promoter. And what that promoter is it's an area that allows for the binding of RNA polymerase. So RNA polymerase is the enzyme that's going to bind to the DNA strand at the TA promoter region. We're kind of ignoring the fact that there's lots of other stuff going on. There's other transcription factors and so on. But once RNA polymerase binds to TA, it's going to read this in this direction, and it's going to form, so it's going to read it in the 3 to 5, which means it's going to make the new strand in the 5 to 3 direction, which is similar to DNA. And then so what we did is, so the RNA polymerase is there, but I'm just not showing it. What we did in class was then we lined up complementary mRNA nucleotides. So like after the TA, I would need a G to be across from the C there. And I would need an A to be across from the T. C across from the G. And then across from the A would be a U for uracil, because RNA does not have thymine. RNA utilizes RNA has some differences between DNA. RNA is single-stranded. RNA uses ribose sugar instead of deoxyribose sugar, just a minor difference in an OH instead of an H on one of the carbons. And I think it's the second carbon. And then also the bases, so uracil instead of thymine. And so then we just continued all along the strand building the RNA. And in this case, what we're building is messenger RNA. So a lot of times I might say mRNA, messenger RNA, because this is going to contain the code for the gene. So the gene is going to start here, and it will go all the way to the end of this. And at some point in the end, there's a stop region, but we'll get to that. And so what I'll do is I'll just kind of show a finished one. Kind of cheat here a little bit. Okay, and so here is a completed messenger RNA. Again, starting with the G, A, C, U, and so on. So we kind of went along and made that whole strand. And we also did some modifications to it. So we added a 5' GTP cap, guanine triphosphate. And we also added a 3' poly A tail. 3' meaning here's the 3' carbon, and so this is the 3' poly A tail. In our body, this would be hundreds of adenines long. We just threw about 8 of them on there. So there's the completed messenger RNA. That's the process of transcription. Transcription is when you form messenger RNA from DNA. And that took us a whole day, because it's a lot of, an hour. It took us a whole hour to cut these out and put them in the right sequence, and then add the GTP cap and the poly A tail. So now we have a finished mRNA. We didn't do exons and introns. We didn't do any splicing of that, but we did end up with a messenger RNA. And then what we did next on the second day is we took our messenger RNA and we translated it into an amino acid sequence. So in order to do that, we used these cards that say amino acid being carried, tRNA, and it has the bases. So I didn't have like specific ones for each of the 20 amino acids, but I did just have these blank cards. And so what we did is we started here. And so what would happen is the ribosome, actually we didn't start there, we started at a start codon. The ribosome is going to be where translation occurs, and that happens in the cytoplasm, right? At the ribosomes are either attached to the endoplasmic reticulum, or the rough ER, or they are just floating around in the cytoplasm. And a ribosome is going to look for a start codon. And so we use page 197 in the book to identify start codon. So if I can zoom in on that section there. Okay, so we use the genetic code, and these are all the mRNA sequences that code for the various proteins, right? And so one of them is the start codon, and that happens to be AUG. Let me see if I can make sure this is focused here. Okay, so here's the AUG start codon, and that codes for methionine, and we're going to get to, at the end of this, a stop codon. So like UAA, UAG, or UGA are mRNA codes that will code for the stop of translation. And all the other ones in between, right, proline, threonine, alanine, and so on, these are going to be in the middle of the protein. Okay, so we used page 197, and so we knew that we were looking for an AUG to start. So as the ribosome is kind of moving along here, here we go. Here's our AUG. That means that that's going to be the start of translation. And so at that AUG spot right there, we wrote the complementary anticodon. A goes to U, U to A, and G to C. And then we wrote the name of the amino acid that this tRNA, transfer RNA, would carry. So again, this is methionine. So I'm just going to write met for methionine there. Okay, and then then students would go on and do the next one, and then every 3 now, right? So starting with the start codon, every 3 nucleotides are going to get a tRNA. So again, we wrote the anticodon on the tRNA, A to U, C to G, U to A. But we don't look up UGA, we look up ACU. We look up the code on page 197 is ACU, because you look up the mRNA code, not the tRNA anticodon. So ACU, look that up. ACU is threonine. So I'm just going to write THR for threonine. And just that process continued. And so rather than having you watch me build all of them, I'll just go ahead and show you a finished product there. Let me zoom out a little bit. So a finished product with all of the tRNAs holding amino acids. This one doesn't have the amino acids cut off of the tRNAs yet, but methionine, threonine, arginine, leucine, aspartic acid, and so on. So this fit all the way down until it got to a stop codon, which actually it doesn't have an amino acid. The stop codon isn't an amino acid. It just meant the student just put this on there just to show that it's the end. So when they got to the end, the code read stop. And so what I had the students do then, if they had time, was then to cut out and remove the amino acids from the tRNAs. That would happen at the ribosome there. And then this is the finished product. So this was the finished product for day 2 when we went through translation at the ribosome. So the finished product for day 2 was an amino acid chain, which is otherwise referred to as a protein. So again, the three things that we started, or the things that we went through, is just a quick summary. We started with the DNA template strand. We then made a messenger RNA strand at the nucleus, that's transcription. And then we transcribed that into an amino acid sequence using tRNAs and the ribosome, and that's translation. So transcription and translation, mRNA from DNA and amino acids from the mRNA. Hopefully that was helpful and made sense. That was the RNA and proteins paper cutout activity.